Probability in Punnett Squares. This is your textbook section 11-2. In this I'm going to be showing you how to do a Punnett Square. Hopefully remember this from 8th grade. So let's get started. You have to remember what probability is. Probability is the likelihood that a particular event will occur. It's like the probability of getting hit by lightning or the probability of winning the lottery. It's not a guarantee that something will occur, and it's not a guarantee that it will not occur. The only guarantees with probability is if there's a 0% probability or a 100% probability. So this is just how likely something is to occur. And if you remember back to, I'm sure in some math class you learned how probability is calculated. Let me come back here. And probability is just the number of things that you want to have happen over the total things that could happen. Now let's practice this just a little bit. Alright, so what is the probability of flipping a two-sided coin so it lands head side up? Well, you just have to make a little ratio here, and we want one event to happen. You divide that by how many things could happen. Two things could happen. So there's a 1 in 2 chance that you could flip a coin and get head side facing up. Let's try another one. Rolling a 4 on a 6-sided die. Well, if there's 6 sides, that's how many things could happen, and you only want one of those things. So you have a 1 in 6 chance of rolling a 4 on a 6-sided die. Take a minute and think about pulling a 9 of diamonds out of a 52 card deck. So we have 52 total things that could happen. You want one card. So you have a 1 in 52 chance of that occurring. It doesn't mean it's going to happen. It doesn't mean it's not going to happen. It means that it's likely not going to happen. Pulling a club card out of a 52 card deck. Well, if there's 52 cards, you put that in the denominator. And the numerator, you put how many things that you want to have happen. Well, there's 13 club cards. So it's 13 out of 52. You can reduce that down a little bit to 1 out of 4. Now, how about this one? Flipping a coin and having it land head side up after the previous 10 flips have all been tails. Well, think about it. What happened before does not affect what's going to happen now. So it's still a 1 out of 2 chance. You want head side up. There's 2 things that could happen. The 10 things that happened before do not influence what's going to happen on that next flip. Each event is independent. And that's something we're going to have to remember as we go through Punnett Squares. Okay, we said last time that for every sexually reproducing organism like humans, there are 2 alleles for every trait. The way those 2 alleles interact determines the organism's genotype. And then we said, why are there 2 alleles? Hopefully remember, one came from Dad. So Dad gave us 1 allele for every single trait through the sperm cell, and Mom gave 1 allele for every single trait through the egg cell. So that's why we have 2 pieces of DNA that we call alleles for every single gene, and those make all of our traits, all of our inherited traits. So if you need to note any of this, take a minute to write it down. Alright, now let's review Punnett Squares. I know that you learned this in 8th grade, but a Punnett Square, all it is a diagram. It's just a tool, and it just shows the combinations of genes that offspring could have when 2 parents mate. That's all it shows. The possible outcomes when you look at one gene from each parent, and we'll take a look at this in just a minute. Whenever I use the word cross, that just means mating of 2 sexually reproducing organisms. So I'm using that word cross a lot, and that's all that I mean. 2 organisms that reproduce sexually, producing offspring. And we'll use a Punnett square to see what alleles their offspring could possibly inherit from Mom and from Dad. Alright, so Whenever we do a Punnett square problem, there always has to be some given information. You always have to know what's going to be dominant and what's going to be recessive. So we'll stick with pea plants just because that's what Mendel had studied. And we'll say the allele for tall height is dominant for the allele for short height. So the way that we note this, if you remember back to 8th grade, is the dominant one we represent with a capital allele, or a capital letter. So here big T is going to stand for dominant, and then short height, we use the same letter, but we're going to use the lowercase version of that. So whenever we need a dominant allele, we're going to use the capital T. Whenever we need a recessive allele, we're going to use the lowercase t. We don't use a T for tall and an S for short. We pick one letter and we stick with it. So we have a male plant that's heterozygous tall and a female plant that's heterozygous short. Actually, let me get rid of this word. This is a mistake. All right, so a male plant that's heterozygous tall. Well, hetero means two different things. And if it's tall, it's got to have a capital T. But its other parent couldn't have given it another capital T because we call that homozygous, because homo means the same. So its genotype has to be big T, little t. And if the female is short, that means that she can't have any tall alleles, because the tall alleles are dominant. And if she had a big T, she'd be tall. So the only way to be short is to be lowercase t, lowercase t. That way she doesn't have any tallness dominant alleles, and that way the female plant can be short. So this is going to be our first step, is taking the word problem and making our genotypes for each parent. Okay, now take a look at the pink, I think it's the pink paper that you have, maybe it's yellow, and this is going to be your guide to Punnett squares. So the step one we kind of already did, but we'll do it again. You have to take the word problem and then figure out what your alleles are going to be and the genotypes for each parent. So we said we're going to cross a male that was heterozygous tall with a female plant that was short. So we're going to pick a letter and stay with it. You can use A's, you could use any letter you want. I just always take the dominant trait and use that first letter. So it'd be heterozygous tall, and tall is dominant. You need one capital T and one lowercase t, because hetero means two different things, and to be tall means you have to have one capital T. To be short, you can't have any capital T's, because if you had one capital T, you wouldn't be short. So that's what that parent's genotypes have to be. So in our notation, we're going to have the one parent is going to be capital T, small t. The other one's going to be small t, small t. So that's step one, taking the word problem, figuring out what's dominant, and then writing what we're told about the parents into these symbols, these genotypes. Remember, one genotype is made of two alleles, one from its mom, one from its dad. Same thing over here. There's the one from its mom, the one from its dad. All right, let me show you step two. Step two, you draw a Punnett square. And what you do with your Punnett square, you take one parent's alleles, doesn't matter which one, and you put them on the top. You put one over top of this column here, and you put the other allele over this column here. Remember, the other parent was small t, so we'll put those over here. Now this part, you take this large T and you copy it down here and down here. So we'll make a big T here and a big T here. And same thing with this little t. We're going to copy it down into this box and down into this box. This little t, we're going to copy it to the right and to the right. So we'll put the small t here from that parent, and we'll put the small t here from this parent. And this small t, we'll copy it here. And we'll take this small t and we'll write it to the right, right over here as well. Now, a lot of people like to stop here and say that they're all done, but this doesn't show me that you know anything. Just is a bunch of t's in a box. So when we do Punnett squares, you can't just stop here, because you need to show me what all of this means. And that's what step 3 is. Step 3 is interpreting the Punnett square. That's you proving to me that you know what all these t's inside of here mean. So remember, here was the one parent's genotype. Here's the other parent's genotype. And what we have in here are 4 possible combinations for their offspring. Okay? So the probability of this one coming out is 1 out of 4. Probability of this one's 1 out of 4. Same for this one, same of this one. None of them are guaranteed, but it's just the probability of each one of these being the offspring. So let's see how we interpret this thing. Okay, over here, I want the genotypes and the probabilities. So if you look up at your Punnett square, we only had two different genotypes inside the box. We had big T, little t, and we had little t, little t. So just write them all out. How many times did big T, little t happen? It was 2 out of the 4 boxes. It was the 2 boxes to the left. And if you'd rather say percentages, I'm writing with a mouse here so you can tell my handwriting isn't great. You see there's a 50% chance of having a pea plant that was big T, little t. Now, what are the probability of having a pea plant that would be little t, little t? Well, it was the 2 boxes to the right. There's a 2 out of 4 chance of that. And so that means there's a 50% chance of having plants with that genotype. Whoops. Okay, so genotype means this, the type of genes, okay? The 2 alleles that you got from one from mom, one from dad. Phenotype means the physical. What are these plants going to look like if we let them grow? So, what would big T, little t look like? Well, if tall is dominant, then these are going to come out tall. So we'll just write the word tall. And again, there's a 2 out of 4 chance that a certain pea plant will be tall, and that is, there's a 50% chance that that pea plant comes out tall. So if we had 100 peas from this cross and we chose one at random, there's a 50% chance that one would grow into a tall plant. Or if we planted all 100, about 50 of them should be tall. There's no guarantees, it's just probability. Now these would come out short because they don't have any of the tall capital T DNA. So these would be short. And because there was a 2 out of 4 chance of that genotype, we have a 2 out of 4 chance they come out short. And again, that is a 50% probability that a certain pea plant from those 2 parents would be short. Okay? And then whenever you do a Punnett square, take a look at the problem. If they're asking you to do anything else, make sure you answer any of the other questions. But those are the 3 basic steps. Taking the word problem, making it into the 2 genotypes, then copying them into your Punnett square, then transferring all the letters down the columns and across the rows, and then here we are at step 3. Interpreting the Punnett square. Tell me what the probabilities are of each genotype and what the probability is of each possible phenotype. And they don't always line up like this. It's not always 50 here, 50 here, and then 50 here, 50 here. Okay, sometimes you could have 3 different genotypes here and only 2 different phenotypes over here. And if you pay attention and take things nice and easy, you'll see what I mean. Now, your homework tonight is or for the weekend, is to go over that SpongeBob genetic sheet. The top part, you just have to look at the phenotypes and tell what the genotypes are. They're going to show you some genotypes. You have to write what SpongeBob and Patrick will look like. They're going to want you to tell them which genotypes are dominant, which ones are recessive, homozygous, heterozygous, things like that. And then there's a few Punnett squares, 2 at the bottom, and there's a few on the back. You're going to have to do them on a separate sheet because you probably won't have enough room. And then we'll go over those on Monday. All right, so please email me if you have any questions, and I'll see you all on Monday.